Welcome to this module on Staphylococcus differentiation. Staphylococcus species are a diverse group of bacteria commonly found on the skin and mucous membrane of both humans and animals. Accurate identification of these species is essential for proper diagnosis, treatment, and understanding of their clinical significance. In this lesson, we will explore the key biochemical tests and characteristics that allow us to distinguish between different Staphylococcus species, that is coagulase positive and coagulase negative, and we will learn about other tests like VP test, Trehalose fermentation, hyaluronase, and beta galactosidase. By the end of this section, you will be able to systematically identify Staphylococcus species based on their biochemical profile and understand their relevance in both clinical and veterinary microbiology. Members of the genus Staphylococcus are gram-positive cocci that typically appear in grape-like clusters under the microscope. These bacteria are commonly present in skin and mucous membrane of humans and animals. While many species are part of the normal microbiota, some are important pathogens responsible for a wide range of infections. Important examples include Staphylococcus aureus, Staphylococcus pseudointermediate, Staphylococcus hycus. Identification of these organisms in laboratory relies on a combination of staining characteristic, hemolysis pattern, biochemical tests such as catalase and coagulase reactions. So let's start the differentiation of Staphylococcus species. In diagnostic microbiology, identification of cocci begins with microscopic examination using gram stain. So the first step will be gram staining. This staining technique allows microbiologists to differentiate bacteria based on the cell wall structure. When the organism appears as gram-positive cocci arranged in clusters as seen in this picture, they are more likely the members of the genus Staphylococcus. This characteristic grape-like cluster arrangement occurs because the cells divide in multiple planes. And some cases we can see gram-positive cocci arranged in chains. Those will be streptococci. In some cases, the gram stain may reveal gram-negative cocci, that is pink staining cocci, instead of gram-positive purple staining organism. These bacteria are not Staphylococcus or Streptococcus. Instead, they commonly belong to the genera Moraxella or Neisseria. These organisms require different biochemical tests and identification method. The second test that we will conduct to differentiate the Staphylococcus species is catalase test. It will help to differentiate Staphylococcus from other gram-positive cocci. Catalase is an enzyme that will break down hydrogen peroxide into water and oxygen, producing visible bubbles. You can see in this slide. If bubbling occurs, the organism is catalase positive, which is very typical for Staphylococcus species. If the organism is catalase negative, which will be more likely to belong to the genus Streptococcus. These organisms can be further classified using Landsfield grouping, that is a Streptococcus group, which detect the specific carbohydrate antigen on the bacterial cell wall, and this classification is commonly performed using coagulatination test. However, there is an important exception within the Staphylococcus genus, that is the organism Staphylococcus aureus subspecies anaerobius, which is belong to the Staphylococcus genus, but this is unusual because it is catalase negative despite belonging to the Staphylococcus. This bacteria remain coagulase positive and typically produce hyaluronase, an enzyme that facilitates tissue invasion. So the second test we have to conduct is catalase test. Catalase positive means Staphylococcus except aureus subspecies anaerobius. If catalase negative, that will belong to the Streptococcus or this Staphylococcus species. So now we learned that most of the Staphylococcus are catalase positive. For catalase positive Staphylococci, the next step is coagulase test, which determines whether the bacterium can clot plasma. Based on this reaction, the Staphylococcus species are divided into coagulase positive Staphylococci and coagulase negative Staphylococci. So in this picture, these two are coagulase test positive that belong to the Staphylococcus aureus, and this is coagulase negative. Coagulase positive organisms such as Staph aureus are usually associated with more severe infections, whereas coagulase negative species like Staph epidermidis are commonly part of the normal skin microbiota but can act as an opportunistic pathogen. Therefore, by systematically applying gram staining, catalase test, coagulase test, and further additional biochemical reaction, we can accurately differentiate Staphylococcus, Streptococcus, and other gram-negative cocci in clinical and veterinary diagnostic laboratories. Now we learn how to differentiate coagulase positive Staphylococcus. Coagulase positive Staphylococcus species can be differentiated first by their ability to produce hemolysis on blood agar. Hemolysis is the breakdown of red blood cells, an important characteristic used in bacterial identification. There are two main types of hemolysis observed with coagulase positive Staphylococcus, that is gamma hemolysis and beta hemolysis. Gamma hemolysis, also known as no hemolysis, which means shows no clear zone around the colonies, indicate the bacteria do not lyse the red blood cells. An example of the species that exhibit gamma hemolysis is Staphylococcus hicus. This species have coagulase negative isolate also. Keep this in mind while differentiating. So Staphylococcus hicus is both coagulase positive and coagulase negative. Next type of hemolysis is beta hemolysis. It is characterized by a clear zone of complete hemolysis surrounding the colonies. Beta hemolytic species include Staphylococcus aureus, Staphylococcus pseudointermedius, and Staphylococcus delphini. Observing the type of hemolysis on blood agar is a crucial step in the laboratory for identifying coagulase positive Staphylococcus and narrowing down the species responsible for infection in both humans and animals. Beta hemolytic species need other biochemical tests to differentiate it separately, will be discussed in the coming slides. Next test we will conduct in the beta hemolytic Staphylococcus species is VP test or acetoin test, which detect the production of acetoin. A positive VP test indicated by the red color change suggests the presence of Staphylococcus aureus, and the VP negative we need to continue testing with additional biochemical assay to differentiate other coagulase positive species. So if a bacterium is coagulase positive, beta hemolytic, and VP test positive means it will be Staph aureus. Next is if it is VP negative, we have to continue testing. Next we will perform trehalose fermentation test. A positive trehalose test shown by the color change in the medium, that is yellow coloration is the positive, which means it is Staphylococcus pseudointermedius, and if it is trehalose test negative, the organism is likely Staphylococcus delphini. So beta hemolytic streptococci, three species are there: Staphylococcus aureus, Staphylococcus pseudointermedius, and Staphylococcus delphini. In that first test we will conduct is VP test. VP test positive, then it is Staph aureus. So all the negative isolate we have to move on with the next test, that is trehalose fermentation. In trehalose test, if it is positive means Staph pseudointermedius, and it is negative means it will be delphini. Next, let's focus on coagulase negative Staphylococcus species, which are commonly found on the skin and mucous membrane. These are opportunistic pathogen. Some of the most frequent species include Staphylococcus epidermidis, chromogen, felis, and hicus. These bacteria share typical characteristics like catalase positive, coagulase negative, and usually exhibit gamma hemolysis, means no hemolysis on blood agar. It is important to note that the Staphylococcus hicus can be somewhat unusual. I already told that earlier. While many isolate are coagulase negative, some strains can be coagulase positive, showing coagulase positive reaction. This variability means careful biochemical testing is essential to accurately identify the species. Understanding these distinctions help in diagnosing infections. Now let's look at the identification of coagulase negative Staphylococcus species. So first test we will conduct will be VP test or acetoin test, which detect the production of acetoin during glucose metabolism. If the VP test is positive, showing a red coloration of the medium, the organism is identified as Staphylococcus epidermidis. And if it is VP test negative, we have to continue the testing to the next test, that is hyaluronase test. This test detects the enzyme hyaluronase, which helps bacteria break down hyaluronic acid in host tissues. If the hyaluronase test is positive, the organism will be Staphylococcus hycus, and if the hyaluronase test is negative, we have to continue for additional testing. So the third test we will conduct in coagulase negative Staphylococcus will be beta-galactosidase test, which determine whether the organism produces the enzyme beta-galactosidase involved in lactose metabolism. If the beta-galactosidase test is positive, that is indicated by the yellow color, the organism will be Staphylococcus felis, and the negative will be Staphylococcus chromogen. By following this step-to-step biochemical testing method, microbiologists can accurately differentiate important coagulase negative Staphylococcus species, which is particularly useful in veterinary diagnostic laboratories.